Thank you everyone for coming. We were expecting quite a few people. I hope that they have the time right. But we do want to get started. So I'm Laura Mattei. I'm the Director of Land Stewardship with Sudbury Valley Trustees. And I'm so glad that all of you are here tonight and enthusiastic about helping to restore pollination systems and native pollinators. Sudbury Valley Trustees, if you're not familiar with us, is a nonprofit organization that protects open space and natural habitat for people and wildlife in 36 communities that surround the Sudbury, Assabet, and Concord rivers. SVT is a member-supported organization, so if you're not a member, we hope you'll become one and support the conservation work that we do in your community. SVT coordinates the activities of the Metro West Conservation Alliance. The Metro West Conservation Alliance is a regional collaboration. It involves land trusts, municipalities in this region working together on conservation. The Native Pollinator Task Force formed in 2018. And SVT has been supporting that effort over these years with dedicated staff time, as well as providing a website space for some of the materials and providing some communication support as well. And last month, the task force has come up with a new name for the effort. It's called the Bumblebee Project, Creating Native Habitat. Now I'm forgetting it. Creating habitat for native pollinators. And the mission of the Bumblebee Project is to conserve native pollination systems by providing the specific habitats required by at-risk pollinators and to reduce the threats to these pollinators. And this work is based primarily on the recommendations of Dr. Gegear, who you'll be hearing from this evening. I would like to introduce the task force members who are here this evening. Karen Paquin, who's our current chair. Karen's from Margot. She also sits on the board of the SVT board. She's the president of our board, in fact. Giancarla Kalpas is from Chelmsford, and she has a wonderful pollinator preservation garden. If you haven't seen it yet, you should go see it. Sam Corbin is our conservation coordinator this year. She's working with Sudbury Valley Trustees, and she's the staff person most dedicated to the effort. And before Mark's not here, and Liza Smith is from Maynard, and she's going to take it over from here and share some current news.
Hi, everybody. The Bumblebee Project and our task force have been very busy. We have updated our website so that it's more streamlined. We have organized a native plant kit sale that's being hosted on the Sudbury Valley Trustees website. You can order the plant kit until the end of this month, and we expect pickups to be happening in early June. We've also been co-hosting community meetings. They've been organized by the amazing and lovely and talented Sam Corbin. Sam's coordinated with local garden clubs in Wayland, Ashland, Bedford, and Framingham to host both virtual and in-person meetings. And in those meetings, participants can learn about the Bumblebee Project and learn how they can get involved in the initiative. The Bumblebee Project would like to see a pollination preservation garden in each of the 36 towns in our region. And so far, we have 12 communities involved in creating these habitats. I'd like to now introduce our honored guest speaker, Dr. Robert Gegear. He'll be speaking for about 45 minutes, and then we'll save your questions afterwards. Dr. Gegear holds a PhD and is an assistant professor of biology at UMASS Dartmouth. He's also the founder of the Ecology Project, which recruits the help of citizen scientists from across the region to digitally collect and submit ecological data on native pollinator species using smartphone and web apps to help determine the cause of wild pollinator decline. Dr. Gegear has been enormously generous with his time and his knowledge, and we feel incredibly lucky and honored to have him with us. Please give a warm welcome to Dr. Robert Gegear.
Thank you very much for those kind words and the invitation and the support. This is my first in-person talk in quite a while, probably for many of you as well, so I need to get adjusted to faces instead of screens that are shut off. So today, as was mentioned, I'm going to talk about some of the work I've been doing over the past 5 years or so on native pollination systems, conservation of native pollination systems. And there are a few things that I want to accomplish today. I think the first thing is to help you to see what the problem is and is not. So giving you some background information about pollination systems. The term pollinator is used quite a bit, and I give quite a few of these talks and workshops, and I ask the audience, well, what is a pollinator? And I have yet to get the proper definition. So that tells me that there's a problem and there's a knowledge gap, and so the first part of my talk is to help to fill in that knowledge gap so you can see my approach, right? So people have different approaches to conservation. They have different reasons for putting in gardens. They might want to put in something showy that is pleasing to them, which is perfectly fine. But if you want to conserve something and conservation or restoration is the goal, there are very specific things that you need to do. So in the case of the bees today, we'll give you the background. I'm going to talk about what I've done and also give you success stories. You'll notice in the title it says more than just the buzz, and that's because this really isn't about bees. Right? Bees are one player, but there's a difference between the bee and its functional role as a pollinator. And the reason that we need to conserve these things is because of its functional role, not the bee itself. It's the interaction, and that means the functional role pollination has to do with native plants. And so what's missed in all of this is that it's really a native plant focus. The bees are helping the native plants, and you have to think of them as a unit, which is why I refer to things as pollination systems and not pollinators. Because people think if they see a bee on a flower, it's a pollinator. Well, I'm telling you, it's not necessarily a pollinator of that plant. And there are many things we see on flowers that aren't actually pollinating it. And we need to be able to pay closer attention to who's actually doing the work, and we'll talk about why that's important. Right? So Beecology Project, the bee is in there, but what people need to focus on is the ecology. A lot of projects for decades have been looking at bees and where they are. That's of interest to me, but not as much of an interest as what those bees are doing. And that's what separates what I'm doing from what everybody else is doing. To figure out what they're doing, we need to understand behavior, we need to understand the interaction with the plants that they're visiting, and also we need to take things from the plant's perspective. Who are the important pollinators? How are those connections made? Right? And I'm hoping that at the end of this, at the very least, if you don't want to help out with the project, and I'd love if you participated in Beecology, you at least understand what the problem is and is not, because there's a lot of misinformation out there, and I hope to cover some of those things today. All right, so we'll start off basically what is a pollinator, right? And we'll build up to pollination system. And so we need to step back, and there are a wide variety of animals visit flowers to feed on the nectar or pollen or both, right? The nectar, we can think of it as their source of fuel. The pollen is a source of protein. Bees can't make more bees without pollen, all right? So it plays a very important role, but bees can't fly around or survive, in the case of a bumblebee, more than 24 hours without nectar. So both of those things are important sources of food, and they need pretty much a continuous supply to persist. Globally, there are about 200,000 of these species. When we look at the variety of species that we see on flowers, the vast majority of them are insects. So of the 200,000, only about 1,000 are vertebrates, like our hummingbird. So we have bats, hummingbirds, small mammals that get a lot of attention because they have a backbone like us, and they're cute. But the bulk of the work is being done by the bulk of the animals, the flower visitors that we see are insects, a wide variety of bees, but again, Beecology says bee, but we've now expanded the project to include butterflies, butterfly pollination systems. I've worked on hummingbird systems, right? They're bees and flies, and a lot of other things that are on flowers are important for that role as pollinators. So you can see in the background, all of these animals are faced with choices, right? If we go out to a meadow, midsummer, there are a lot of different things in bloom. And so how is it that these animals figure out where to go? There is a widespread view is that they're using innate preferences for colors. So bees like purple and moths like white. And in reality, there are very, very few bees that are that highly specialized, that are just relying on innate programming. So what they're doing is they learn the cues associated with the nectar or pollen rewards, the quality of those rewards, and they track them over time and space. So there's a lot going on in their little brains that we need to consider. And when we hit them with pesticides, it affects their ability to make these good decisions, which has implications for the plants, which we'll talk about. So how do we know that this is the case? Well, what we can do is bring it into the lab and test it. So everything that I do is based on the collection of data and science, the scientific method. Here we have a monarch butterfly. Put it in a harness. We can let it go after this. It's naive, meaning that I raised it in the lab. It hasn't ever seen a flower before. So if you touch the legs of the butterfly with sucrose solution, so that was a cotton swab soaked with 20% sucrose solution, it sticks out its tongue like Pavlov and Pavlov's dogs with the saliva. So it sticks out its tongue as soon as it starts to ingest the nectar. So what we can do is show it a color, we can show it an odor, we can show it a shape, combinations, to try to figure out whether it has a learned preference for those stimuli. So in this case, we showed yellow. The tongue didn't come out until I touched it with the swab with sucrose. Once the tongue touches the sucrose swab, it forms an association between that color and reward. And so we can test it. There you see there's no movement. The proboscis or tongue of the butterfly is not moving. Antennae aren't moving. We show a color very similar to the test color. Still no response. And then we show the butterfly the test color eventually, and we get a fairly rapid response where that butterfly is starting to look for reward. So if I give it another color with less of a reward, then if I gave it a choice and was flying around, it would take the one with the higher reward. We can do this with bees. I've done this with a wide variety of different animals. And so they're learning and remembering, and what this tells us is that we need to make sure that we're giving them good sources of nectar, that it's not just about the color. We really need to think about the reward that that plant's offering and make sure that we have what it needs. What we're finding, and I have a grad student working on this, is it isn't just about the sucrose, that there are plant volatiles in the nectar that they're attracted to for whatever reason, possibly to help keep them suppress infection. So there's more to it than just the sugar, amino acids. There's a lot of other things in the nectar as well, and as well in the pollen. All right. For some reason, this is not working. So if we look out, so they're using learning. Now, imagine there are all these species competing. They have different behavioral tricks that they've evolved, or different morphological features that help them to outcompete those animals. So they're all competing for a limited resource, and you're going to get winners and losers. And so through natural selection, they've evolved different ways to outcompete others. So here we have a goldenrod on the left. We can see Bombus impatiens is just poking around. The nectar is exposed. Very easy to get to, very easy to find. That's why we see a lot of different things on goldenrod. Now that Bombus impatiens has a medium tongue, that's good for short tongue species, but there are other species like Bombus fervidus that has a long tongue. And Bombus fervidus doesn't do well on goldenrod because its tongue gets in the way. Bombus fervidus has evolved a long tongue to exploit and has co-evolved with these tubular flowers like this Impatiens capensis flower, this jewelweed flower. You see the spike here. The nectar is located at the base of the spike, and Bombus fervidus sticks its head in and wraps its tongue around to get the nectar. So there's a good fit. Our short-tongued bees, unless they're nectar robbers, can't go in legitimately, go in the flower to get that nectar because their tongue's too short. So it's like there's no nectar for those short-tongued species. For Bombus fervidus, when it goes over here, it can't compete, so it's like this isn't an option for Bombus fervidus. So we are already starting to see that species are separated, and also that the individual species are paired up with plants. So this is all looking at things from the animal's perspective and foraging. They're trying to maximize the amount of energetic gain, maximize the amount of pollen they're bringing in per unit time, in the case of bumblebees or social species, to bring to the colony to make more bees. So now we switch things. As those pollinators, as those flower visitors at this point are moving from flower to flower and competing with one another to collect nectar and pollen, if and only if they transfer pollen from one flower to another, and that transfer leads to fertilization and the production of seed, then and only then does its name change from flower visitor to pollinator. And this is the problem. People see things, and I'll show you some examples. They see animals, bees, flying all around on plants, and they think, oh, I'm helping the pollinators, but they're not pollinating anything. And those bees can't pollinate until you give them a plant that they actually pollinate. I mean, it's subtle, but it's extremely important that you understand that difference. And when I ask what's a pollinator, most people say it's an animal that transfers pollen. Well, that's not the proper definition. Why? Because there are many cases where bees transfer pollen and other things transfer pollen, and it doesn't lead to fertilization or the production of seeds and fruit. So there's nothing going on in terms of plant reproduction. So these flowering plants, the vast majority of flowering plants, require an animal to transfer their pollen to the surface of a stigma, and we get the pollen tube growing down, we get fertilization taking place. So if a plant, so we have an inflorescence here with many flowers on it, if we had a bee or a butterfly or something that's collecting nectar, moving up and transferring pollen, a lot of plants are what's called self-incompatible. That means that they can't receive their own pollen, and they can't get fertilized by their own pollen. So we've got tons of pollen transfer occurring here, but there's no fertilization, no production of seed. Some plants are self-compatible, so the transfer of pollen would lead to the production of seed, but often the seed quality, because they're mating with themselves, so that's going to reduce the ability of those seeds that they produce to survive. So from the plant's perspective, they want the animal to move from one plant to another, and this process is called outcrossing, right? We want to maximize our genetic variation. That helps our offspring to respond to any sort of environmental change or a variable environment. We also have the situation where they pick up pollen here and they transfer it to another species. So we have pollen transfer, but we're not going to get any pollination taking place, right, because we're transferring our pollen to a different species. So what this does, two things. We're obviously wasting our male gametes. Right? The pollen's being wasted here. And also, when this species receives foreign pollen, it blocks the surface of the stigma. And so this plant cannot receive, or is limited in its ability to receive its own pollen to get fertilized. So pollen transfer by itself does not mean pollinator. We need to know that it's helping the plant to reproduce. So, for example, right, here we have obedient plant. We have Bombus fervidus here and Bombus affinis down here. Bombus fervidus is going into the flower to get the nectar, making contact with male and female reproductive organs and leading to fertilization. So here we have Bombus fervidus that's a pollinator. So we can classify it as Bombus fervidus as a pollinator of this plant. Here we have Bombus affinis. Bombus affinis, its trick to compete with other flower visitors is to bite holes at the base of tubular flowers. It's a nectar robber. There's another one, Bombus tricola. This one is no longer in the state, but Bombus tricola is in Western Mass. They do the same thing. So they can't compete. They have a really short tongue. They can't compete with long tongues, so they cheat, right? Our Bombus affinis is a flower visitor. It's not a pollinator, right? And not only is it not a pollinator, but we can consider it a parasite because it's taking the nectar and not giving the plant anything in return, no benefit in return. In fact, when our Bombus affinis takes the nectar, our Bombus fervidus comes on the scene and says, hey, there's no nectar here. I'm going to ignore this plant. So it's actually deterring the major pollinator. And plants have evolved a variety of mechanisms to deter this from taking place. But what's interesting is that the poster child for pollinator decline is Bombus affinis. But it is not a good pollinator of tubular flowers because it's robbing the nectar. So I could have thousands of Bombus affinis on my obedient plant and still have no pollinators. I'm not helping the pollinators. I can't say that until I introduce a plant that Bombus affinis actually pollinates. And if we never introduce that plant, we can't hope that it's going to be a pollinator. We have to know that we're putting something in that it's going to actually pollinate. And this is something that's lost on people as well. Fine, if you have a species that's blooming later or another species in the area that Bombus affinis is going to visit and pollinate, and it's just getting nectar from this plant. But if it's not there, then we've lost that functional role as a pollinator. Similarly, I could put a feeder in here and replace this with a feeder or a non-native plant and keep thousands of Bombus fervidus, which is also a species in decline, keep it going. And this is protecting, I'm saving the bee, but I am absolutely losing its functional role as a pollinator because I don't have the plant. So if I could invent a feeder to feed all the bees in trouble, I'm doing a disservice because I've saved one species, and as we'll see, when you save their functional role as a pollinator, you're saving hundreds of species. You're protecting ecosystems and systems rather than a single species. That is the importance of these animals visiting flowers and pollinating. That's the true importance of a pollinator. And the reason that things get confusing is because when this started, it was with honeybees. Honeybees are non-native bees, and they're important, very important for pollinating crop plants. And so when people use the term pollinator, it was inferred that they meant pollinating crop plants. We need these animals so they can help us to pollinate crop plants, and 2 out of every 3 bites of food we take is based on a pollinator, or one of these bees. The other butterflies and other things aren't important in that context, or as important. But that's, I think, where things got confusing, and people started to apply flower visitor, and now, oh, it's a pollinator. But forgetting about, in the case of native plants, that you have to have that native plant there that it's paired with, otherwise you've lost its functional role as a pollinator. So if we look at the plant and all of the animals that contribute to its pollination, that's what we're calling a pollination system. So what I want is moving from thinking about the bee to thinking about the bee-plant combination, that interaction as a unit. We don't separate the two. We treat them as a unit from this point going forward. And things can get very complicated. So here's some data from my grad student, Chelsea Hettering, on penstemon. All right, so this case, penstemon digitalis. And what, if you visited Breakneck Hill at all, or Lincoln, you'll see these bags over flowers, so that was her experiment. So what we were looking at was, first of all, what's the importance of animal pollination to penstemon, and also, what's the importance of specific pollinator species that are considered of conservation concern to the pollination of this plant species. And so some of the flowers were bagged, and we just saw how much pollination occurred with no animals. We then looked at, opened up, took the bags off and just let anything visit the flowers for a week or two to look at open pollination. And then the third treatment, and it's difficult to see the X down here. This one is, we opened up the bag and the first visitor, we let one individual visit. We recorded who the individual was, what they were doing, and then we rebagged the flower to see what contribution a single visit or a couple of visits, how that would contribute to pollination. And what you can see is that very clearly, so this is the fruit weight. So fruit weight, this line is showing you the fruit weight you need to actually produce seeds. And you can see that very clearly this plant needs animal pollination, so animals are important. But what this shows you is that there's a lot of variation based on the visitor. So if you have a small bee that goes in and doesn't make contact with the male and female reproductive organs, we're not getting those seeds produced. We have Bombus vagans that goes in and is large enough and fuzzy enough to make contact. We see that it's actually a really good pollinator of penstemon. So there's variation. We've got flower visitors down here. We've got the pollinators here, and we can use this method to separate out and figure out who are the major pollinators of these native plants. And this becomes important when we switch this out for Penstemon hirsutus, which is a plant species of conservation concern. And she's got data on Penstemon hirsutus, and it's not quite ready for presentations yet, but we're seeing the same pattern, but we can figure out who are the important pollinators of this plant species of conservation concern, and then we can figure out if that's a contributing factor to the decline of those species. Plant species. So they're complicated and they're also diverse. So there are plants, again, we're flipping to the plant's perspective. Some plants are looking for short-tongued bees, small bees, large bees, long-tongued animals. Some plant species are targeting hummingbirds. Some plant species are even targeting flies. Right? So we get a lot of focus on bees, but flies, again, the importance of the animal is relative to the plant species. So this plant species, it's a one-to-one. If it doesn't have this fly species, the plant species goes extinct. Similarly, if this plant species goes extinct or declines, this fly species is also going extinct. It's a one-to-one. Other systems are more generalized. So we lose some important ones, but we still have some less efficient pollinators to fill the gap. Butterflies are important. Here, all the specks you see on this butterfly, that's all pollen. They're transferring pollen well. They're all contributing to some extent. As I mentioned, so this one's pollinated by hummingbird. Notice where they're placing the pollen. Here, the pollen is placed on the forehead of the hummingbird. Why? Because if the plant places it on the wings, it's not a very efficient way to transfer pollen, to maximize the plant's reproductive success. Bees will visit this. They can get to the nectar, which is located down here. But in doing so, long-tongued bees can get to it. In doing so, they bypass the reproductive organs. Again, so they're not contributing to the pollination of this plant. And the plants have evolved mechanisms to deter bees from visiting them and then encourage the hummingbirds. So there's a push-pull that's going on. So when you put in a plant, there are a bunch of species that are going to come to that plant, but there are an equal number that are going to be deterred because they don't like that resource. So this one-size-fits-all approach, which we seem to have, we're starting to move to look at species and what the species needs, both from the animal side and from the plant side of the equation. So why is this important? I mentioned agriculture. Those systems, it's a handful of bees, honeybees non-native, it's extremely important, pollinating a handful of plants that aren't native. They may have been, but we've selectively bred them to maximize output for our benefit. So it's like a few species to a few species. When we look at things from the ecological perspective and this diversity of native pollination systems, what it is the foundation of ecosystems, right? So you can imagine, in the case of agriculture, the pollination is producing fruit and seeds and things for us to eat. Ecologically speaking, this diversity of systems is providing a diversity of food, nest sites, habitat, shelter for a wide variety of species at other trophic levels. So you can imagine that we get our pollination taking place, these pollination products, so seeds and fruit produced are feeding small mammals and birds, and those small mammals and birds are food for predatory species. So as we start to erode the foundation, we remove these systems, we're removing the diversity and abundance of food available to this trophic level is going to be increased competition. And at some point, we're going to affect the ability of this level to survive and reproduce, which is going to have a cascading effect through the ecosystem. So if we remove or reduce the diversity here, we're going to have what's called ecosystem collapse eventually, where we get this massive reduction in biodiversity, because everything is connected together with those pollination systems and the diversity of those systems being the ground level for all of this. And this is important for not just, so why is all this diversity important? And the answer is that it's important because of what are ecosystem services. So these are things we get from nature for free. So, carbon sequestration, soil decomposition, water purification, all of this comes from healthy, diverse, functioning ecosystems. That is the true importance of pollination and pollinators from an ecological context, and it takes diversity. So there are only about 5%, I'm being generous, 5% of our native bees actually pollinate crop plants. So if we're going to develop conservation strategies to help save the bees, we're helping 5%, and we're completely ignoring the vast majority of species that we have, and certainly those species like Bombus affinis that are very close to extinction or local extinction are going to be ignored because it's a numbers game in agriculture. You just need one thing to pollinate acres and acres of one thing, and once you get that done, you're good. This, you have to keep the diversity to keep the diversity and keep that system going, all right? And that's the difference between those two. This isn't getting any attention. It's getting a bit more now, thanks to a lot of people in this room. The agriculture is getting all the attention because it's obviously helping us economically and also putting food on our table. But this, through the ecosystem services, eventually is going to affect us. When we get ecosystem collapse, we're going to have to figure out a way to replace these services, and many of them we can't replace, and if we do, there's a hefty price tag associated with it. So it's better to protect it than to figure out what to do once it's gone, because as they say, extinction is forever. All right, so this is to hammer the point home. If we want to build biodiversity, right, we got to what this idea that we focus on the species, so bee, butterfly, whatever it is, versus its functional or the system. What does that mean in terms of conserving species or impacting species? And I have this example, and everybody feels bad at the end and cringes and say, oh, do I have to take down my hummingbird feeders? And all of this is informed decisions. I give you information, you do what you want with it, right? But so here we have a hummingbird feeder. We have lots of happy hummingbirds flying all over. We're happy too. We get to see them outside of our window. Then we have over here, we have Monarda, which is pollinated primarily by hummingbirds. And so if you're a hummingbird and you've got a choice between a patch of Monarda and a hummingbird feeder, there's the phrase bird brain, but birds are actually extremely smart. These hummingbirds are going to go one-stop shopping. They fill up, they go back to their nest. They take cover, so they're not going to be affected by a predator. To get the same amount here, they'd have to visit, 100 flowers, and move and be exposed more. So there's an increased risk of predation. So given the choice, they're going to take this, which is reasonable from a foraging economics perspective. So that's great. We save the hummingbirds, but the pollination of this plant and the persistence of this plant population, it's a host plant, right? So at the larval stage for the orange mint moth, so at the larval stage, we've got a lot of birds that use the larval stage of butterflies and moths as a major source of food. If you've heard Doug Talmy speak, that's how he intersects with what I'm doing. He's focused on feeding birds, and I'm focused more on the pollination system, but it's the same idea that we've got these native plants that are host plants to native moths and butterflies. Then the adult phase is going to feed our eastern whippoorwill, right? So we've got our purple finch that's going to eat the seeds. So we've got all these species that are benefiting from the products of pollination. So if I put in a patch of Monarda, and it doesn't have to be that large to support a mating pair of hummingbirds, I get all of this benefit, diversity-wise, versus putting up a feeder and I help one thing versus helping several things, right? Same thing if we put in non-native species. So remember, everything has co-evolved to fit together, and that goes through the whole system. Everything is intertwined and interdependent. When we put in a non-native species, even though this hummingbird may pollinate this species, and it may be a good source of nectar, the birds don't want to eat the seeds. The butterflies and moths don't want to use it as a host plant, right? So we're cutting out all of this by doing this, even if we do get pollination. And that's why we need to shift to native plants, because we know that they've co-evolved, and that's what they like, because they've evolved all these different tricks to exploit them. From the plant side, you put in a native plant against this, it's going to pull the hummingbird away from the native plant, so there's competition from the plant side for pollinators, because they're limited as well, and we're not going to get the pollination of our Monarda because this has more nectar. All right? It comes down to the economics. All right. So hopefully at this point you know what a pollination system is and what I'm focused on. So it's the interaction between the animal and the plant. That interaction is what I'm focused on. So what's the issue? The issue is that, as I said, these things are being degraded at an alarming rate worldwide. So this isn't a Massachusetts issue. This isn't a North America issue. This is a global problem that we're losing connections. So here are some data from bumblebees. So I looked at historical records, and I went back to those same locations and surveyed to see what's there. Between 2015 and 2019, I continue to visit these sites. I just haven't updated. But you can see that there are many species where the yellow bars are lower than the blue bars, indicating that there's some sort of decline. In the case of Bombus affinis, there is no yellow bar because that one's likely locally extinct, although I still have hope. Butterflies, so this is new. So I was able to get the data from the Mass Butterfly Club through a publication, and I analyzed data from 2010 to 2018 to try to figure out which species are in decline and which ones are stable and which ones are increasing. And this is the ones in red here showing you all the species that are considered in decline in Massachusetts. And then we flip things over, and we're seeing the same thing in our native plants. Many species are declining. Not surprisingly, many of those species in decline, like Penstemon hirsutus, are pollinated by a lot of the bees or butterflies that are in decline, so we're losing those systems. So clearly there's an issue here. But what I want to point out, and what people don't realize, is that not all species are in trouble. So again, save the bees. Look at, there are many bee species where the yellow bars are higher than the blue bars, and that's because a bee like Bombus impatiens bumblebee species is expanding its range, and it's much higher and much more abundant than it was historically. So we see some, and that's true with the butterflies, and that's also true for native plants. So we have these differential declines. So if we go out and we see a bunch of bees and we think, oh, look, I've got lots of bees, I'm doing a great job. Well, if it's all these bee species, and for the most part it is, that are doing quite well, that are common and have increased in abundance or are stable, there's no conservation needed. So we need, again, to shift and target the species that are in decline, both from the animal side and the plant side, and figure things out. And so how do we figure them out? Data. We need to collect data. So, back in the day, people liked to go out and they'd learn about the natural history of species. Funding agencies didn't like that. They wanted more hypothesis-driven results that could be applied. And so we're in this mess because we're not investing in the ecological data that we need to understand what these species need, and then how are we affecting what they need. And that's what I'm trying to do, is to fill some of these knowledge gaps. So for butterflies, they need a host plant, and they also need a source of nectar. So the butterfly gardens that I see, it's great, you've got a lot of nectar plants, but if you don't have host plants that they like in the area, you're not going to get that butterfly species, right? So for bees, you know, we'll focus on the foraging side, the nectar and pollen, but they need a place to overwinter. So right now, all these bee species are overwintering, and we need a good place for them to overwinter, which is why you don't blow your leaves. And I'll get to that at the end of the talk. They come out, they're going to need a place to nest, right? And then once they get a place to nest, they need that pollen to make new bees and the nectar to keep the bees that are there going. If they don't have a nest site, if it's a bumblebee that doesn't find a nest site, the queen lives for a month and then dies. And that stops the population dead in its tracks, right? If all the queens died this spring, that species would be locally extinct in one season. So we need to think about what the needs. From the plant side, as I mentioned, who are the major pollinators of these plants? How much do those plants depend on seed production for that population to persist? These are questions that need to be answered. And so, as I said, to answer those questions, we need to collect data. In the case of bumblebees, you know, I've been working on bumblebees for 30 years plus now. You know, obviously they have a place in my heart, but they're not the only thing that's important. There are many other bees and butterflies and everything else that's important. But to start, what I noticed was I was working on monarch butterflies migration when I was at UMASS medical school, and I was watching my advisor at the time, I couldn't even use the word bee in the lab, so it was very restrictive. So once I got out of that situation, I was following the bee decline issue, and I knew that a lot of species were in decline during my PhD. I knew Aphanus was in decline at the time, and I was trying to figure out what people are doing, and I noticed that there were a lot of plant lists. Plant this to save the bees. And I looked at the list, and I realized that Bombus fervidus, or Bombus aphanus, didn't really visit the species that were on these lists. And so, you know, knowing this is one thing, but getting data to convince somebody that this is the case is something else. And so what I did was I started to go out and collect data. So here are the initial study sets I had, some in Eastern Mass, some in Western Mass, and surveyed bumblebee flower interactions from May through October, and looked at all the data over multiple years to figure out what these different bees need. I looked at queens, males versus workers in bumblebees, and looking at species level differences. That was a very difficult, you know, I'm one person, I had one grad student, and all these sites, these initial surveys that I did showed very clear results that these species are doing different things and like different things. So to expand the project, you know, I had my grad student, I needed to get data quickly, and so I decided to recruit citizen scientists to help me to collect data. So early on, they would send me pictures, then I realized videos were better, they'd send me short videos. Then to go through all those videos was a time-consuming process, so I paired up with Liz Ryder and Carolina Ruiz at Worcester Polytechnic Institute, and they developed the phone app and the website with information. And so the citizen scientist in the Beecology project was born. You can go out with take a video of a bee interacting with a flower, and then the app will help you to identify the bee species, the plant species, and importantly, the behavior. Was it collecting nectar or pollen? What exactly was it doing on that flower? And so I crowdsourced the data collection. And so here's the Beecology web app, and so it's a web app, meaning you go to the website and you don't need to download it from the App Store. So as long as you have a functioning web browser, it doesn't matter if you have an iPhone or Android, you can participate in the project. But through that, so in parallel, so I had citizen scientists, and I appreciate all the beecologists that are out there and future beecologists a great deal. At the same time, I was collecting my own data. All right, so here are some data that I had from just one site in July, and here are the bumblebee species down here. The ones in red are the target species, so those are the species of conservation concern. And you can see if we divide them up based on tongue length that they're doing very different things. The long-tongued bees, not surprisingly, are all over vetch and red clover, which have a long tongue. a long tube, I should say. We've got Turnarius and Terricola, which have a really short tongue. They're on the milkweed and on goldenrod and things like that, Joe Pye weed. And so that wasn't surprising to me. But what's surprising is when you look within these groups, Bombus vagans loves Prunella vulgaris. So we're seeing these species-level preferences within the long tongue or medium tongue category. Bombus impatiens, late in August, everything you see is probably going to be a Bombus impatiens. They're doing something very different. They're on white clover, for example. None of these other medium tongue bees are all over white clover to the extent that impatiens is. So we're seeing these differences. But the great part of this was when you look at the beecology data, the same patterns emerged. So this is people just going out in their backyards or they're going on a nature walk collecting data, and the data that we see line up with the data that I found through my primary research, very controlled, rigorous experiments where we walk transects and control for temperature and a variety of other things. And so from that, collecting the data from the Beecology project and also from my primary research, I was able to put together a list of plants. So if, let's say I wanted to support Bombus fervidus through its life cycle, right? So they're coming out in the spring and they're persisting through early fall. We need to make sure they have that nectar and that pollen. One of the biggest findings was that there are pollen sources. So certain plants, these bees love for pollen. Like that's their pollen source. And so those are down here, just to give you an example. And then they're using different plants for nectar. So here are some nectar plants from the spring, pedicularis all the way through to the fall, jewelweed. This assemblage would support those two bumblebee species of conservation concern through their life cycle. So last year we added a butterfly component to the app. So now we're starting to collect butterfly flower interaction data. We teamed up with iNaturalist, so we have the iNaturalist API that we pay for, so it will help to ID the butterfly, the flower, and we're starting to collect data there. We've identified the target species, as I mentioned, through the Mass Butterfly Club data up to 2018. And my grad student, Annie Monroe, has been out the last 2 years collecting butterfly data, the same way we collected the bee data, and we're finding the same patterns. Butterfly species have preferences. So within skipper butterflies, certain skippers are doing one thing, the non-native skippers are doing something completely different, and we're seeing these species-level preferences. So here we have the common ringlet. So we looked at, were they visiting long tube flowers, native, non-native, what were the preferences for these butterflies? So just looking at tube length, the common ringlet, only 3% of the 2,000, 3,000 observations that we have in general. So the common ringlet was less. Let's say it's 100. 3% of those were to flowers with a long tube. So there's a very clear short tube preference versus 50% for the little glassy wing. And then we go down to the silver-spotted skipper, and we see that 90% of the flowers had a long tube. We also saw a lot of preferences for native versus non-native. Some species seem to prefer native plants, some seem to prefer non-native plants. Some species, like the eastern tail blue, all of the observations we have were visits nectaring on pea family plants. And so we're seeing different levels of preference, and a lot of those species of conservation concern that we identified have these specific preferences. So if we're going to add things to our backyard or to conservation land and we're trying to target those species, they, just like the bumblebees, have very specific needs, and all the other bees and everything else out there at the species level are going to have these needs. So the next phase is to expand it to non-bombus bees, and I'm certain that we're going to get certain preferences there as well, and we're trying to figure out what the nature of the preferences are. So that's exciting new data. So if we're trying to maximize our systems, two plant species came out clearly on top. So looking at the conservation value of native plants, Prunella vulgaris and Monarda officiosa, we're supporting all the bumblebee species of conservation concern, plus a bunch of butterfly species of conservation concern. And Prunella vulgaris, even though we didn't have a lot of it, it was supporting a lot of different bee and butterfly species, and I'll show you an example in a second. But in case of bumblebees, so I was surveying Lincoln, a property in Lincoln, and there was a small patch with maybe 4 or 5 Prunella vulgaris plants and tons of acres and acres of other things, and I only found Bombus vagans on the Prunella. I didn't see it anywhere else. That's how strong some of these preferences are. So obviously, if I'm trying to save Bombus vagans and I'm putting in something that it doesn't prefer, I'm missing the target completely. Bombus fervidus has such a preference for red clover. That same field, there was a patch of red clover. I saw a couple Bombus fervidus workers. They mowed it. Obviously, I told them not to do it, but they did it anyway. And this wasn't Lincoln Land Trust. This was whoever owns the property. No Bombus fervidus were observed on site for the rest of the season after that mowing. So they are very highly dependent on red clover, but when you give them natives, they shift over to the natives, and Prunella being one that they really like, or Monarda, or the penstemons. Right. And so feeding the bees red clover, protecting or conserving or restoring pollination systems, we put in the native plants that they like and that they're pollinating. All right, so species level conservation at home, just to, again, give you some examples. This is my front yard. You can see it's, you know, I let it grow out, so this is sort of speaking to the Nomo May. Nomo May, we let it grow through May, we get lots of white clover and dandelion, right? We do that, we see none of the target species. We see European skippers, but no native skippers. So what I did was I threw down some Prunella seed. You can see some here. And once that came into bloom, I let this go. This has honeybees, has lots of activity, don't get me wrong. Lots of honeybees, lots of Bombus impatiens. All those common bees and non-native skipper butterflies are all over this white clover. This comes into bloom. These are pictures. Here's Bombus vagans visiting in my yard, and here's some skippers. So I've got my entire property is white clover. I've got a few patches of Prunella, and that's where these animals are showing up. I don't see them on any of this white clover. All right. So it's not just about nectar being available. It's do they like the nectar source for whatever reason. So this has led to the plant list I have on my website. I have not updated the website. I need to include the butterfly target list. I need to update some of the plants to reflect those changes. So that's coming. I mean, the 2022 list isn't terrible, but once I find the time, I'm going to update before things come out. And with the warm weather, things might be coming out sooner than I expect, so I better get on this. But that's coming. And I'll make a note on the website that it's a 2023 edition. But again, the observations that go into this list, you can't see it because it's cut off for some reason. But at this point, I have 20,000 observations going into this list. All right? So it's, you know, I can go into an area and see what's there and tell you what I expect to see at this point. And as we switch to, okay, let's put these plants in and see what happens, there are very obvious benefits or positive results that we're going to see. So the Beecology Project and everybody involved in the Beecology Project, tremendous help with data collection. We need to see if this actually works. So during COVID, I switched from, I have enough data, so let's start getting these plants and putting them into areas and see if we can support these target species. So we started with the bumblebee species, and we're expanding this year out into the butterflies. And so this is getting the citizen scientists to become scientists, become more of the scientific process, not just data collection, but hypothesis testing. So going into areas, seeing what's there, making a change, and then seeing what impact that change has made. It's very important to me you don't just trust me. I don't want you to trust me. I want you to put these, take a chance and put these plants in, but I want you to see that there's a positive effect. And you can see that the target species was not there, and you put in the plant, and now it is there. Because that's going to empower you, and you're going to spread the word, and it's a much more effective way to do this than for you to just blindly trust me, right? And so I give a lot of talks. I'm happy to give workshops in this area. I've given a number of workshops on how do you identify these bees and butterflies, so you know what to look for, and you can look at whether they're pollinating or not. We can learn about plant-pollinator interactions, so you know what to look for, and then you can take those tools and go home and then do the science for yourself, backyard science. So to start, though, I have properties, and again, if you want to, help out and put some of these plants in, I can use that as a study site. I would greatly appreciate it. Again, everything I do is data collection. So this phase where we put in these plants and see what impact it made, I have primary sites in Dartmouth, in Southborough, Breakneck Hill is my first site and my primary site, I would say. So I've got a lot of support from Freddie and others at Southborough, which is great. Rehoboth, I've got a major site, and I know people here are putting in plants, and I plan to visit everybody and do data collection and do a walk and talk or whatever it takes, so I really appreciate that. But you can see, I mean, it's hard to see if we went back to the map of Southborough that Breakneck Hill is 40 acres. This is 260 square meters, and Breakneck proper is like 20,000. So we're not talking about a huge change, but in all cases where I put in these plants, the targets showed up in the first year. So we're putting in plugs and plants, they bloom, the species are there. So to me, this was shocking how rapidly these bees that weren't there before or were on red clover in low numbers are now on the plants that we put in. And so this was 2 years ago. I'm seeing increases in the number of these species every year. So that, even if they're there in greater numbers than they were the year before. And if this trend continues, we're going to really get a healthy population that we're supporting of these target species, and also the native plants we put in that they prefer are getting pollinated and providing food, shelter, nest sites to everything else that's in the area. So making those connections that I talked about. So just quickly, and often it's not putting things in, it's removing things. So here at Breakneck, they had a major purple loosestrife problem. And on purple loosestrife, didn't see any of the target bumblebee species, saw tons of honeybees and bombus impatiens all over, large numbers of both of these. So removed the purple loosestrife. The year after it was removed, all of a sudden, all this mimulus, so these are natural populations of mimulus, where a huge population showed up where the loosestrife was no longer. Also, all these other native species show up when they weren't there before. So what loosestrife does is it locks up the native diversity, native plant diversity. So you get rid of it, you've got all these native plants that I'm telling people to put in. If you just control the loosestrife, you're going to have them available to these things. You're going to restore connections that way. And of course, and this is a video. This video shows on one mimulus plant, I had both of these species. Some years I go through Breakneck, 40 acres, I don't see a single Fervidus. Here I saw Fervidus and vagans on the same plant during the same observation period, which is not typical at all. All right, so the other thing that I want to point out is, there's a lot of talk about honeybees on conservation lands, and so I want to give you an example of why it's not necessarily a good idea. All right, so these are some data that I collected. So this is Monarda rubra that was, and so this is showing you the last week of July, so 2 weeks worth of data collection. So last week of July down here, and then the first week of August. So the last week of July, both of the target species were all over. Love this Monarda rubra, which is why it's on the list. But we also saw a low number of carpenter bees. So carpenter bees love to bite holes. They have a short tongue. They're robbing the nectar. What that does is allows species with a short tongue to invade the system. And that's exactly what happened. And it was honeybees in very large numbers coming in and what's called secondary robbing the rubra. That week, both of the target species were no longer present. They had moved off. I didn't see them. I assume they're on red clover, but that's competition, right? Nectar and pollen are limited. If there's competition, you're going to get winners and losers. In this case, the sheer number of honeybees, 10,000 individuals in a single box, hive, flooding a system, there's no nectar left for these longer tongue. Like they're leveling, this bee is leveling the playing field, and because it's native, they're in low number, that's a part of the system, and these two can still persist. But when this comes in, it upsets everything. All right? So that's just one example of how honeybees can come in. But when we look at things like mountain mint, the numbers are ridiculous. 100 honeybees to 12 the common bumblebees, which are all over the place. So it's like 10 to 1 honeybees versus other things. It's just, and again, From an agricultural perspective, it's ideal because you've got thousands of individuals that can fully pollinate an acre of crop. But when it comes to this and the competition part, that's where we need to think more about where we're putting the honeybees. All right, goldenrod, same deal. Goldenrod, you look at a goldenrod, it's all honeybees, and you'll see one or two other bees. Certainly none of the species of conservation concern are on mountain mint, and this is just reinforcing things like mountain mint, black-eyed Susan, that people like to put in. You're helping lots of honeybees, wasps, and bombus impatiens, but you're not helping any of the species of conservation concern. They're avoiding it completely. So abundance versus diversity, we need to think about that from a conservation perspective. So the last thing, and I apologize, I'm going to have to speed through these last two slides, but this, again, new data looking at the impact. So here's Beecology down here. Here are two pollinator gardens, quote unquote pollinator gardens, that were put in before I came on the scene, and then we've got 25,000 square meters of other things, right? Vetch, red clover. So they were supporting 11 of the target butterfly species and 2 of the target bumblebees, but all of this was non-native plants. In this small area, I'm able to support 8 butterfly species and both bumblebee species in good numbers. So 8 of the target butterfly species, so species I consider of conservation concern. These other pollinator gardens, the same size, but because they don't have the right plant composition, are supporting 3 and 2 of the target butterfly species. So this is supporting those 2 plus 6 more, and is almost equivalent to 25,000 square meters versus 200 square meters, right? So it's, it doesn't take a lot. I don't need to change 40 acres. I just need to put in the right things, and I'm going to have that positive impact. Okay, so this is just an example of what you can do in your backyard. All these plants were blooming at the same time. I think this was late June, early July in my backyard, and I live very close to Garden in the Woods, go there all the time, don't see any bombus vagans or see them in low number. All of these are pictures of bombus vagans in my backyard after putting in these plants, and I didn't see them before putting in these plants because it's all dandelion and white clover. So again, it's going out and seeing for yourself. The other thing is, and this is true at a small scale or large scale, selective mowing. So the neighbors are upset if you have an unkept lawn. If they don't know what you're doing, they'll complain to somebody and they'll tell you to cut it. But here I've mowed everything. Here is prunella that I left. So I mowed around the prunella. I put the prunella at the base of trees. I mowed around it, and I'm keeping that beneficial resource while I'm keeping the aesthetic of my lawn up. I also don't fill in my lawn because here, and this is from my front yard, here are nests for ground nesting bees. So you have a little bit of space. It looks terrible, but it's supporting that diversity that we need those nest sites. So grasses are just as important as the flowers. They act as host plants. Birds love native grass seed, and also things like to nest in native grasses, and those nests are being used by bumblebees. The things that create those nests, the rodents, are being used by birds, like owls. Most of what an owl eats or is running around in an open field. And so we need to think about the connections between those native plants and the seed and the bees that are pollinating to feed the small mammals, like all those connections that I spoke about. And finally, so the nest sites I mentioned. The one thing, and it's a bit late now, but people always ask me about spring cleanup, fall cleanup. Is there a good time? The answer to that is no. And the reason is because those stems, so when you deadhead, you're removing the food, right? If you think of it that way. You cut things down to the ground, things are nesting and overwintering in those stems. We blow the leaves, but those leaves are protecting things that dig into the ground to overwinter. And so you can imagine, so we don't have much snow, as much as we used to. That leaf layer is, so they dig down, so you want a nice soft, you don't want hard pack, you want a nice loamy soft soil, because that's what bumblebee queens and other things are digging down a foot or two. And then the leaf layer protects them from those cold temperatures. Wood piles. There are things that are overwintering in these wood piles. Everything that's messy, that's what they're using to overwinter. And that's why when you blow the leaves, cold temperatures like what we had a couple weeks ago would kill a lot of what's there. You know, so it's giving them a bit of a chance. So we need to think about that. Finally, I talked about connections, and this is again from Breakneck Hill. Here we have a target bumblebee species visiting shrubby St. John's wort. This same plant, we have this white-throated sparrow feeding on the seeds. White-throated sparrow is a bird species of conservation concern. And so what we're seeing are these benefits of putting in native plants and the benefits of the pollination. We're helping 2 species of conservation concern. And so we're going to start to collect data on animal-plant interactions, like the wildlife-plant interactions. Which species are we supporting? Who's using the seeds? Who's nesting? So for this bird species, they want cover and they want seeds close to the ground. That's what they like. And shrubby St. John's wort gives it to them, so that's a source of food for them. And then finally, this is purple giant hyssop. And this was like this in Beecology through the whole season with birds feeding, but all the movement you see, there are probably a dozen birds all feeding on that purple giant hyssop seed, right? Major source of food. We didn't see that at Breakneck Hill until this happened. So if I cut down that purple giant hyssop to clean things up, those birds wouldn't have a food source, right? So those connections we need to think about as well. And with that, I'm sorry for going over time. I'd like to take any questions, and thank you for your time. Thank you. So one last thing. So a lot of the plant species that I have on my list. So Freddie Gillespie, this is the 2nd year. This has been a tremendous success. So they collect seeds and then they do a winter sow, and people are putting these plants in their backyards. I guess there's a lot of seed packets still available, so if you wanted to contact them to get some of these seeds, then that would be great. All right, thank you.